Hello, everyone, and wishing you all a happy new year. Welcome to this year's First Sets online webinar. And before we get started with today's session, I'd like to thank our sponsorship from IAS, which allows us to offer all of these resources free of charge. Make sure to check out our website for more info on upcoming events and meetings and to see everything available for the community. Today's talk is by Dr. Or Dialik, a senior researcher at the Israel Oceanographic and Limnological Research. He graduated with a combined MSc and PhD program in Geology and Environmental Sciences from the Ben Gurion University of the Negev, Beersheba, Israel. He then completed his postdoctoral research associate positions in the Weizmann Institute, Princeton University, the University of Haifa, Hamburg University, and the University of Munster. He has also been the Marie Curie Research Fellow at the University of Malta. His research focuses on the sedimentology, geochemistry, and reconstructing past environment using marine carbonate rocks. His talk today explores the ambiguities in interpreting carbonate facies and the challenges in interpreting these paleo environments using the carbonate rocks and the insights that can nevertheless be gained from studying these rocks. And with this short introduction, I hand over the forum to you.
Thank you, Shraddha. Good, whatever it is for you, wherever you are. Welcome to this talk. Firstly, a huge thanks to Joanna and Stephen and Shraddha and Teresa and the rest of the Sets Online team for having me. And I'm going to be touching about some fundamental aspects of carbonate sedimentology in this talk, particularly facies interpretation. But first, what actually is facies? So this term has been around in geology for about 200 years. First put into this context by Grassley working on the Alps to describe a body of rock with some specific characteristics. As we're moving forward in time, this definition has somewhat expanded, and by the time we're getting to Walter, it's already used to insinuate depositional conditions. So a sedimentary facies is a distinctive rock unit that forms under certain conditions of sedimentation. Going back over 100 years forward to the Flügel book, when he's talking about microfacies, he described a microfacies is the total of sedimentological and paleontological data which can be described and classified from fin sections, peels, and whatever. We have this a huge set of tools and descriptive and interpretive aspect we ascribe to these things, to this thing called a facies. This term is not uniquely to carbonate sedimentologists. It's a term that is also that's been used in our community, but also been described for seismic interpretation and also for seafloor units. And sometimes, like in this paper I'll talk about later, all in the same context. And while these other two are coming from subdiscipline, they all converge to the same aim, to delineate a unit, to delineate little geological unit with common character and possibly an underlying genesis. This is where things get a bit more complicated, because the delineation of facies by itself has never been straightforward. It kind of depends on the scale we're working, the tools we're working with, and similarly, the interpretation of it has never been simple or straightforward. They both depend on the information we can get from the samples and the aims we have when we're approaching the sample set. The ability to infer information is depending on what we can actually extract. And then to inverse it. And before I'm going forward on this, I want to put in a few words about my background, because this kind of colors the way I'm approaching all of this. By training, I am a classically trained carbonate sedimentology. I worked on outcrops, I worked on boreholes, I worked with fin sections, hand samples, that sort of stuff. But as my career progressed, I pivoted to work more and more on modern environments and work more with biologists and chemists. And most of the work I'm doing right now is more environmental chemistry, but it still intersects. And that kind of forces me to look at what we're doing from a very holistic perspective. And with that perspective, I've sort of developed a minor gripe with one of the key perspective of the discipline, because a lot of the way we're looking at this for historical reasons has to do with reef as our main lens, especially euphotic reefs. Reefs are kind of the prima ballerina of carbonate sedimentology. It's where a lot of our focus is. But there are so many other carbonate production environments which make the bulk mass of the stage, and they don't get as much audience attention. If we go back to the classic Wilson model, two of the belts that Wilson delineates have to do with reef. Of course, the organic buildup, but also the fore slope, and to a minor extent, the winnowed sand. So out of the 7 belts, 3 have to do with reef. But of course, our environmental definitions can extend a lot further and cover more environments. Now, I'm not saying reefs are not important. Reefs are very important because, as far as we can see, both in the modern and in the geological past, reefs are biodiversity hotspots, especially tropical shallow water reefs are very, very important. They show higher richness, lower dominance, across right about every period in time. And the tropical shelves themselves are very, very important. They account for the bulk, almost half of all modern carbonate accumulation. But of those tropical reefs, those tropical shelves, reef don't make up that much area. If we go to the Great Barrier Reef, we go to the Northeast Australian shelf, only about 9% of the area is strictly reefs. So what's everything else? It's a bit easier to look at this stuff with outcrops because seafloor mapping and detailed sampling across large area of the seafloor is very, very expensive and very time consuming. And this is something I have been looking at with Giovanni Coletti, whose a name is going to come up repeatedly through this talk because we have been collaborating on a lot of this stuff quite extensively. He and I, together with some students a few years back, started looking at this. We compiled as many outcrop descriptions as we could for the Cenozoic across the Middle East and Central Asia, and built an extensive database describing what makes what builds the bulk of the carbonate and also the other parts. The way we went about this was through a quite extensive data mining operation. We basically hunted down each and every reference we could find which had facies description, preferably with fin section images, and columnar sections. And in each locality, we basically mapped out how much of each section was each facies. And we went into description of the individual facies and delineated what are the main components for these facies and what are the secondary components and how much volume of the section they made up for. In this case, what is highlighted in this purplish pink is facies 6, which was a large benthic foraminifera dominated facies. Now, there were at least two other large benthic foraminifera dominant facies in this section. And we kind of had to start lumping things together, because when you start working across very large periods of time and wide areas, you are kind of forced into generalization, because otherwise the nuances make it impossible to see the bigger pictures. And the reason I was showing that one is because large benthic foraminifera facies are actually quite significantly present across the entire region and through the entire period. A bit more in the Eocene, but across 144 sections we looked at, the main, we found over half of them in some cases were dominated by large benthic foraminifera. And this is kind of interesting and perplexing originally. We knew that through the Paleogene, large benthic foraminifera were quite common, quite dominant. But it becomes very stark when you compare this to what's going on with the reefs. So on the left side, I have a reanalysis of the Paleoreef database from Küstling and Flügel. And on the right side, I have an analysis of our data for the same time slices. This is just for the Eocene. And you can see the foraminifera are present. They make this light blue color on the left side, but they're not the most dominant thing. The most dominant thing across this entire data set are corals. Whereas if we look at our compilation, large benthic foraminifera dominate facies make for the bulk of the volume. Corals aren't that dominant. And this is not something that was just in the Eocene. Across the entire system we looked at through most of the Cenozoic, while in the Paleoreef database, corals make for 50% of the reefs through this period. In our data set, they don't make up for even or 10%. When we have something and corals, they're a bit more abundant in some cases, especially in the Oligocene, but not that much. And we need to take into account that our database might even be a little bit biased towards these environments, because because reefs tend to be a bit more studied, a bit more oversampled for carbonate sedimentologists. They get preserved a bit better. So the fact that we're seeing so little coral abundance suggests that what we're seeing is probably real. What else we found is it has to do with coralline algae. So encrusting red algae. They're not actually very dominant as the principal component in the section, but we did find quite a bit of them co-dominating. So something N red calcareous algae. So that's actually quite important for binding and lithification, because we know that the coralline algae can come in between the grains and bind them together. So that kind of suggests that their binding role might be increasing their significance. So they're not just forming grains, they're actually preserving a lot of other grains on the site. The end result we had from this analysis is that carbonate production, the bulk of carbonate production, even in warm shallow seas, is not just reefs. It has to do a lot more with what's happening in the fields and meadows that are in between the reefs. And while large benthic foraminifera have an outsized roles, and part of it might have to do with their redisposition. This kind of started making us focus a bit more on what actually is making the bulk of the facies, what's making the bulk of the volume, and what are the conditions and controls on that distribution. Knowing what the dominant elements in a facies are doesn't mean we have a good control. Just because we were able to say, oh, we have a lot of these things making our carbonate rocks, doesn't necessarily mean we can say, oh, we had a lot of this environment. Going back to the paper I showed some snippets from earlier, this is a study I carried out in Malta where we had actually excellent habitat mapping. The entire Maltese shelf has been very detailed mapped with high resolution acoustics, verified with dives, and we also had access to AUV photomosaics of the seafloor. So we had a lot of data. I also had wave data, so I was able to delineate the wave zones. Here I'm defining the base, the fair weather wave base, as the 19th percentile of where waves interact with the seafloor, and storm weather wave base as where there's no surface wave effect on the seafloor anymore. These delineations are a bit arbitrary because honestly, these terms are sort of meaningless. This is something that Peter and Luce already pointed out in 2012 in their very nice geology paper. They analyzed wave data from across the US Atlantic coast and Caribbean coast. And they basically illustrated that there's no segmentation between fair weather and deep water. It's and storm waves. It's a continuum of wave distributions. If we still want to use this term, fair weather wave base, we need to come up with new and clearer definition. I suggest using 19th percentile, and that would put the fair weather wave base in the Mediterranean around 25 meters, 50 meters in the Caribbean, and 100 meters in the East Atlantic. This is something I want to develop further, by the way, so if anybody is interested, talk to me later. And it's also very important to note that waves and seafloor features are not directly linked. This is from a new paper that just came out last week. In the Eastern Mediterranean, again, very good wave data from local buoys. And we're seeing these very nice, very large bed forms made of gravels, way deeper than where the waves are getting to. This is because we have other mechanisms affecting grain distribution, including internal waves, including bottom currents. But if we would see these sort of features, these very large dunes with gravel in an outcrop, we can very easily mistake them from being shallow waters, maybe even coastal, where actually this is a mesophotic reef. So this is something to keep in the back of our head. But let's go back to Malta. Now, in Malta, we had the benefit to do something we can't really do with outcrops. We had the facies. We know what's on the seafloor. We have it imaged. So we could go from the environment and the control mechanisms over to the facies and calibrate them. Or at least that's what I was hoping to do. The problem was that once we started working fully quantitative here and take the components, start putting them with various environmental parameters, one on top of the others, it was kind of hard to find a very clear control. Distance from shore here, it does factor in, but if you look at the distribution in these ordination analysis, and what these are is essentially dimensional compressions where we take all the different grains, each of them plotting them against one another and kind of squishing everything into two-dimensional projection. And we look at the data points. So the blue ones are the sandflats and the bad forms. The green ones are the seagrass meadows, and the red ones is what we would call merle. So these are fields of red algae nodules. We don't really see a clear relationship between the modern observed facies and the facies component. They're actually kind of mixed together. So the seagrass and the sandflats cluster together, and some of the sandflats cluster together with the merle beds. And they do form a gradient. So we have a very clear gradient, which is, again, dominated by distance from shore and water depth, which converge here, as well as possibly some other factors. But we're not getting in the microfacies the ecotopes we're seeing on the seafloor. This is becoming a problem because this means that our ability to invert is suboptimal and maybe not even true. But that, it's one location. It's weird. It's in the Mediterranean, which itself has quite unique systems. Surely, if we look at the entire planet, at all the shelf system, we can find and generalize some rules we could use to better constrain and calibrate facies models, like light. So this is a map of light zonation across the entire planet. The blue line delineates the base of the top of the euphotic zone. The red and orange delineate the base of the euphotic zone, depending on the definition you prefer. So light is very important. We can probably find some relation and regulation by light and temperature and the rest of the abiotic parameters we can look at. That rationale drove Giovanni and me down quite a deep rabbit hole. We still haven't came out of, and worse, we started pulling people in with us. We began kind of obsessively collect and digitize every bit of shallow water assemblage and facies data from the modern seafloor we could get our hands on. Some were easier. Herbert Gischler, for example, does amazing work in the low latitudes, and he's very good about publishing his data. I wish more people would take the way he works as a model. Others, well, let's just say we spent a lot of time digitizing graphs and tables. Some were old, like 70s, 80s. That makes sense. Some were very recent. But by 2023, we had about 3,000 data points. Well, we identified 3,000 data points for which any work was done. In practice, we were able to quantify the components only at about 1,500 of these localities. Those are the green points on the map. But that's already quite a nice data set with quantitative description. This is, like I said, by the way, we're still working on this, and this is our current map. Our student, Gulia Klemparti, has been working and expanding this, and now we have almost 4,000 data points in the database, and continue to work both because we were able to locate more data from gray literature and because people have been sharing information with us. So expect some more stuff in the upcoming years. Maybe in 2-3 years, Julia can be here and talk about her work. And what we've been also starting is looking more into the relationship between hydrodynamics and biochemical parameters. That's still ongoing work. I hope we can get that out soon, but it's been proving a bit more tricky than we originally hoped. But for each of these data points, we have information about what's there. So here I plotted up where and how much coralline algae we have, how much corals we have, and we can look at how they relate to one another. So just for fun, here's a cross plot of the abundance of coralline algae versus corals, which scatters but kind of shows this inverse relationship. Well, that's cool and that's good, and that gives us first control on how we build our facies, but we want to figure out what's controlling them. And for that, we needed to get environmental parameters to control these observations. For what we did back then, I didn't want to go into the models, mostly because of resolution issues. And what I really wanted was continuous data sampled the same way across a long time series so we can get long-term averages. So you're not dealing with just the effects of a single season of where people were out there in campaign. And also don't need to account for differences in years, but have a robust, statistically sound, representative parameters. The way we went about this was to take satellite data. We were able to construct a 30 years satellite time series, extract for each locality all the parameters that we had in the various satellite data. So for example, here I'm showing the chlorophyll concentration in the water column from MODIS. We also had information on temperature, information about light flux. We had water depth, latitude, bunch of other things, and we were able to calculate how much radiation is getting into the seafloor, that sort of stuff. Just for the record, all the data we generated for this study and all the scripts are available online with redundancy. We have them in multiple repositories. And there's documentation of the workflow and all the analysis that we did, so you can go and dig into this if you feel like it. Now, so we have all these points where we have the facies, and we have the abiotic parameters, and that kind of allows us to do something really cool. We can set aside all our a priori assumptions. We can look at this tabula rasa. Here is what makes what we find, and this is the setting in which we find it. And try to build a generalization of the controlling parameters from that. Just looking at the distribution, we do see some trends we recognize. So for example, Zooxanthella colonial corals, marked here as ZCC, are very common in tropics. They make actually for a lot of the assemblage in the tropics. They're weighing down as we go into higher latitudes, and by the time we get to the cold latitudes, they're gone, which that fits our textbooks. On the other hand, filter feeders aren't very common in the tropics, become more common in the temperate, and by the time we get to the higher latitudes, they're very abundant. These are marked as SBFF here. And there are the things that are actually everywhere, like red coralline algae, marked here as RCA. We find them in all the latitudes. Mollusk, marked here as MOL, similarly abundant across everywhere. But this is not granular enough. This is not what we need to really generate these clear rules governing the facies. We need proper multivariate analysis. And it has to be multivariate because one of the things we noticed when we correlated the various parameters is that the correlations for individual parameters are not very good. They're actually quite bad. The only thing that showed some strong correlations were are prima donnas, the colonial corals. So these are the SBCC here on the allochem chart, and they show correlation coefficient of higher than 0.5 to latitude, to amount of radiation in the seafloor, to temperature, and that sort of stuff. But this is why it's important to plot your data and not just look at the correlation coefficients, because once we plotted the data, it looked like this. And that basically told us that it's fine that we have a correlation, it's kind of meaningless. Once we moved to multivariate space, actually, let me stop here. Let me walk you through what's going on here because it's a lot. On the top here, we have all the data points. They're colored for visualization, not doing any delineation here. And similarly, these groups are delineated based on latitude just for illustrative purposes. The bright ones are shallow waters, and the further you go towards purple, you go into deeper waters. And similarly, the rounds are tropics, then subtropics, temperate, and the triangles are the higher latitudes. The lines are the vectors for the abiotic variables. We got temperatures, that marks as SST, and radiation at the seafloor, that's SW on one side. Then we have the extinction coefficient, chlorophyll abundance, latitude, and water depth on the other side. And a lot of the explained variance here is on the x-axis. So, and that's kind of where we see this smearing. On the bottom, each one of those orange dots represents the components we have in the assemblage. So HAL for Halimeda, for example, SPFF, sisal benthic filter feeders. And one of the things that was already quite troublesome is that we don't really see any separation. On the left side of the diagram, we see these carbonate factories that are dominated by corals, by Halimeda, coralline algae, mostly from the low latitudes. This is what Schlager would have called a T-type factory. As on the right side, we have assemblages that are dominated by mobile arthropods, echinoderms, sessile finger feeders, what we would call a C-type factory. But there's no separation. This is a gradient across from one to the other. This brings me to one of the take-home message that I already want to put out there. And that's carbonate facies are essentially segments along an ecological gradient. Using a different analysis and projection here, I think this becomes much clearer. We have this wide ecospace growing from warm, oligotrophic, shallow, high light availability, dominated by phototrophs, all the way over to this high turbidity, high nutrient cold and deep. What's governing these axes very much is energy availability and light and temperature. It's all forms, but both of these are essentially forms of energy that affects the metabolics of the organisms. But the other thing that you probably have already noticed is that I'm not separating cold and deep because they converge. This means, by the way, that in theory, if we want to say we have this section. It goes from this coral-rich photozoan facies into this red coralline algae-dominated facies and into a serpulid facies. One could be say it's a sea level rise. That's a valid model, but it could also be effect of a cooling trend, and it can also be the effect of a eutrophication trend. And we'll get back to this paper a little bit later. This means we need to start thinking a bit more like ecologists on facies interpretation. Less trying to fit things into boxes and more looking at things as gradients. Because carbonate facies are expression of energy availability. And I can already hear some of you grumbling in the background, even though you have your mic turned off. And you go like, well, we can't use this on paleo. We have aragonite loss, the information is incomplete. This is not transferable. So let's have a look at that. To test this out, we need a case study with a lot of data, well-established strong abiotic gradients, lots of carbonates, and preferably not too old, so we won't need to deal with evolution and changes in ocean chemistry. As it happens, the late Miocene Mediterranean, just before the Messinian salinity crisis, tick all these boxes. Well, almost all of them. There are a lot of papers and section descriptions. We know that there is restriction because there is a single inlet in the west. And going to the east, we're developing a very strong gradient, something akin to what we're seeing today, but probably a bit more extreme, at least with salinity. And we are very hopeful. We were stacking the deck. We are going to get this working up and running, no problem. But once we actually collected all the papers, we realized that many of them don't actually have that much information. Some of them don't have section description, some of them don't have facies description. Some of them note that the outcrop is there and some things are in there. And we were optimistic. We were going, fine. We have statistical tools to work with presence absence. This is common. We collected all the data from across the Mediterranean, generated a matrix, and we were seeing vectors. We were seeing how things were relating to each other, but statistically, we couldn't see any separation between the sites. Everything was sitting right on top of each other. And the more we were reading these papers, the more worried we became that we're actually missing information in there. There were things that we know were in some of these outcrops, like example crabs, but they were not described in the texts. So we were going like, how many of the zeros we are having, and this is something we also had a problem with the modern, but we couldn't do the data reduction that we could do in the modern data on the Miocene data. We couldn't say how many of these zeros are actually real zeros. So, after the microscope, we collected samples, got samples from colleagues, tormented a couple of students to do point counting, and generated detailed quantitative data for a few sections. We were able to get and analyze so far 5 sections. 2 from the Eastern Mediterranean, 1 in Cyprus, 1 in the Eratosthenes Seamount, 2 from the Central Mediterranean, Aquabona in northern Italy and Salento in southern Italy, and 1 from the Western Mediterranean, from Sorbos, and a shout out to Jesus Reolid for helping us with that one. And just by the way, all of these analyses that I'm showing, if you want to get into that and you want to do them yourself, Emilia Jarchowska and I wrote a paper a couple of years ago explaining how to use this multivariate analysis approach in sedimentology with detailed workflows, and Amelia wrote some amazing demonstrative codes that you can download and use as your starting point. But back to the data. I'm going to focus on the NMDS, which stands for non-metric multidimensional scaling. I think it shows the data the best. And we're seeing differentiation now. We can see that in the green, we have the Sorbos Basin in the south, in the Western Mediterranean. That's on the lower left corner of the field. And we have the Eastern Mediterranean, Eratosthenes, Cyprus on the upper left, on the upper right corner of the field. Right. We're beginning to see a gradient across the basin. One of the interesting things we were seeing in this data set is that the phototroph and mixotroph actually split. So unlike in the modern, where we see them usually converging, here we're seeing them going in different directions. So on one side, we have the large benthic foraminifera and the green calcareous algae. And on the other side, we have the calcareous corals, the encrusting benthic foraminifera, and the red calcareous algae. And this axis of separation is perpendicular to the separation we're seeing between the sites. And when we look at the components and we take what we know about their habitat and draw that out, we're seeing that a lot of this separation is sitting on a vector that's very different than the separation we're seeing. It has warm oligotrophic and salty waters on one side and less oligotrophic fresh and cold water on the other side. And these less oligotrophic settings do have more filter feeders like serpulid and biozoan. The more oligotrophic has more of the algae and corals. And that's great. The biochemical energy that we've seen generating these gradients in the modern is also, we can also delineate that in the Miocene. But these vectors don't account for all the variability we're seeing. Yes, we have the lateral variability with geographical position, but then we have all this spread within the sample set. And when I take these individual outcrops and I do just, again, this is mostly for illustrative purposes, we do this analysis on the individual section, we see that they form their own internal gradients. And that is because when we're looking at sections, we aren't looking at a snapshot in time. We're looking at a time series. And that time series integrates over a period. And across that period, the environment begins to change. And as the environment changes, that doesn't necessarily change the large-scale gradient, but it does form a temporal gradient, which we're seeing manifested in some of this internal variability. So yes, we can see this energy control in ancient sections. Going further back in time would still be quite challenging because we need to develop a new concept for which will allow us to infer the ecological functions at a sense, or at least the feeding habit of the various groups, and classify and reclassify the assemblage based on that. And that's in the future. And something to keep in mind, the way we're looking at this and the way we're interpreting this is a model. And all models are wrong. Some of them just happen to be useful. This doesn't mean that other models aren't. Faces are at the end of the day models. It's a way for us to delineate a large set of data into segments and assign information and assign an interpretation to those segments. We can discretize those segments further down. We can look at them further as gradient, which requires us to work more quantitatively. And we just need to remember at the back of our head what is going on in reality, that our segmentation is a model. So what I want you to take home from this is that interpretation of carbonate facies and microfaces has to be careful and conscientious of the non-uniqueness of facies. We see that temperature, we see that latitude and depth converge. And those can be temporal changes and they can be spatial changes, especially when we look at very, very long time series in places where it crossed multiple latitudes, like Northwest Australia, for example. Quantitative work really helps with further differentiation and inferring of gradients. We don't actually have to go and segment. We can look at this as a continuum. Because carbonate faces, the way we're working, or historically been working, are segments along this ecological and abiotic gradients. They're not necessarily real discrete entities, even though we kind of think of them like this because they might be in separate beds. But these delineations can sometimes be a bit arbitrary or a bit fuzzy, especially once we consider diagenesis and effect and the way that beds are forming. The governing factors of carbonate faces at the end of the day are the factors that are generating these gradients. I think that metabolic energy and metabolically available energy, like temperature, is really a strong component here. To remember the famous saying from James Knoll, carbonates are not made, they're born, which means that there is biology fundamentally affecting the way these faces are generated. And we need to keep this in mind. We need to be conscientious of this. How does all of this relate to hydrodynamics is really something that we need to put more work on, because looking at this from the biogeochemical and abiotic parameters and physical parameters that are not currents is something we can do, but it gives us only part of the pictures. And this is something that we need to continue working on this as a community. And with that, I'll bring it to a wrap. Thank you very much. And Sharda, we have some questions, I guess.
Yes, we do. Thank you for the great, interesting talk. We actually do have questions, but I'd like to remind everyone again that the chat is open for questions, so you can already add them in the chat box and I'll read it out. And I would also like to remind you that you can also add where you are coming from and your name while you're typing your questions. With this, I'll start with the first question we have from Asghar Ali. I could not find any data from Indian plate in this presentation. And why is that? I mean, he couldn't find it. Why is that?
Let me go back to our map. We did have data from India. Not a lot of it, but you can see here in the southwest corner, we do have some green data points. And in the new compilation, we added a few more. We have found reports from northwestern India, but those did not have detailed information that we can quantify. If you have data from the Indian margin or from Sri Lanka, send us an email. We would be happy to incorporate that further into the database. Right now, most of our data from that region is actually from the Maldives and Sharda, I apologize. I forgot the name of the island group to the south.
The
So,
Lakshadweep.
yes, the
Yeah,
Lakshadweep Island. Thank you very much.
sure. I hope that answered the question. And then we have the next question from Oana from Colorado. She thank you for the great presentation and interesting data. And the question is, do you think traditional carbonate facies models still have predictive value, or should they mainly be treated as conceptual frameworks that help interpretation rather than precise predictive models?
I really didn't want to answer that question. But I do think traditional facies models do have a place. They have their role. I am very hesitant about inferring any predictive capacity to them, not because they don't work, because we know they do work, but we need to be careful about them, because if we just assume Walter's law is applying on these, then that kind of puts us into a pitfall, because if we're looking at a period where the changes are not just lateral, they also have a component related to climate change or runoff, which is also a factor, then even though we're not changing necessarily then we're not moving left and right, we're moving in that we have also another dimension of that factors in there. And that's something to keep in mind.
Sure. Yes. I do not see any more questions, but I actually do have a question, and I'm not sure how much it can be answered. But then my question was, so how much of it is, I mean, how much of the paleo environment interpretation can also change with the change in the preservation bias of each of the carbonate factories? For example, you might have the algae being less resistant, and due to hydrodynamics, they would be less preserved, and would that lead to a change in the interpretation of the paleo environments, do you think?
Yeah, this is a big problem. This is something that we're still trying to wrap our minds around. This is why for the mycene we actually focused on near reef environments, because the solid framework offered us a little bit of protection, but preservation would change the assemblage. We know that it reduces the representation of aragonitic components, especially fin-walled organisms. We know that some organisms, like theropods, we are just completely gone. They don't leave any trace. And that means that we are looking at when we're looking at a paleo assemblage on this perspective, it's like looking at one of these alt CRT TV when the antenna wasn't well aligned. So we have a lot of static in the background, and it's obscuring part of our image. That said, if we manage to, if we're very careful, I think we can establish some amount of data that couldn't allow us to not necessarily account, but accommodate for the information loss. And that data can come from also not necessarily just the components. It can also come from things like morphological parameters and size parameters, which provide us with some additional information. I think geochemistry is going to be something we need to incorporate more into our models. And when I say geochemistry, I don't mean carbon and oxygen isotopes, because I feel like those aren't necessarily having optimal fidelity, especially oxygen isotopes, but more looking at elemental data, things that do have better provenance, better fidelity, and could allow us to look at the environmental information and supplement what we're seeing just under the microscope.
Mhm. Sure. There are a couple more questions from Aminu Alamin Abdullahi. Again, there's no place, but is it safe to say that to get a better understanding of carbonate facies, we need to look at the ecology more?
Yes. Short version is yes. We need to look. We need to look at this both on the modern ecology, but also the paleoecology, and it has to be in the wider context of the outcrop. It's not just what's going on in the samples, but what is the context that we have around us, and if we can, and if possible, I would actually say that having more lateral sampling might be something that's very important down the line to really building this ecological framework for the paleo. And in the modern, yes, we need to think how to integrate some more ecological modeling and ecological data. That said, there is a need for us to figure out the way ecology is done from a taxonomic perspective might not necessarily be optimal for us, because there's a lot of things that we know. When we did the global compilation, we had to ignore a lot of ecological data because there are a good amount of seafloor survey that are done with diving, and they have a lot of components that go into them that we don't care about, and a lot of components that we care about that don't go into them. So, for example, large benthic foraminifera a lot of time don't get counted because of the resolution of the way they're scanning. Whereas a lot of the seafloor coverage could be things like flashy algae that just don't get preserved. So we need to incorporate the ecology, but we need to do this in a way that fits our needs. And to remember that we're looking at an aggregated signal. When you're going with ecologists and you're doing surveys, you're looking at what's going on at that particular point in time. And even if you have a time series, that's an integration of 10, 15 years, if a very, very good ecological time series. The integration we're looking at is much longer. So on the Maltese shelf, for example, we were seeing a lot of Halimeda. But when I was talking to the ecologist in Malta, they're saying, yeah, Halimeda has mostly been gone for the last 20 years. So we're integrating maybe for a century.
Sure. Okay, we have another question from Noor from Jordan. She thanks you for the nice presentation. Could you clarify whether differences in measurement accuracy and uncertainty among the various data sources were assessed and incorporated into the results?
And so we spent a lot of time on data evaluation and data reduction when we were doing these various studies. There is a lot of inaccuracy in the original data because the way people work vary quite dramatically. Unlike the biologists that have like these community standards for marine geology, especially carbonate marine geology, we don't have standards. So people worked on different size fractions. They worked on different methods. Some of them worked with insection, some of them worked with microscopy, and they worked with at different taxonomic resolution. So the way we had to deal with this was to do a lot of generalization and data reduction and be very, very careful in the way we are incorporating data. In some cases, we ended up not using data because we thought that the accuracy of the reporting was not good enough to be comparable. And in other cases, we actually had to merge things together because of the way reporting was done. So I think like one of our biggest problems with the current database is foraminifera, because some people separated between planktonic, small benthic, and large benthic foraminifera. Other people just reported foraminifera. And we know that this is a big problem because with large benthic foraminifera, we have the mixotrophs, which have endosymbionts. We have the ones that don't, and in smaller benthic foraminifera, we also have a lot of other behaviors, and that brings in some amount of error into our data set, which we can't reduce. In other cases, we have also issues of underreporting. So mobile arthropods, so that means crabs, lobster, that sort of stuff, not everybody reported them. And we don't know if they didn't report them because they weren't there, or they didn't report them because they didn't report them. And that forced us to actually lump a lot of groups together in order to achieve categories that were meaningful. And in the background of all of this, we were thinking about ecological functions when we were lumping things together. So we might have two groups that are taxonomically separated, but functionally do something very, very similar. And that was one of the solutions we utilized in order to account for that. We're very careful about the way we're phrasing this, but we're starting to think about these groups in the sense of guilds, that they have similar behaviors, they have similar feeding habits, they have similar energy sources, and that allows us to lump them in a way that's more significant and a way that's potentially down the line transferable backwards in time, because we can generate guild level classification even for things that are extinct, because we have some ideas about their life habit.
Thanks. And we have one more question. You're probably aware of the live dead agreement studies, such as done by Susan Kidwell's team. We know that living assemblages of organisms have defined niches, but once the organisms die, they get distributed all around the basin. However, this blurring effect is different in different environments. Do you think this still exists a discrete sediment facies in specific environments where this effect is weak, such as disoxic basins or sites with very high sedimentation rate? In other words, do facies exist as discrete categories but just don't get preserved in pedestrian situations? This is from Amelia.
Yeah, and that's an excellent question. Yeah, this is something we looked inside the discrete data sets that we put into the assemblage, trying to look at some of this. And I have been looking at this actually in situ in several locations where we had very good seafloor mapping paired with directed sampling into the mappable facies. And when we look at that like that, we do see that in certain locations, the living assemblage is a bit more I'm hesitant to say discrete, but more clearly distributed. But even in those places, once we, for example, where we had a continuous seafloor imaging across transects, and we also had box scores from those locations, and we looked at what's going on in the box scores versus what we were seeing in the videos, we do see that there is some smearing, that the boundaries are fuzzier, even for the living assemblage. There are locations where if we go into very high taxonomic level, then we do see something that resembles discretization. So for example, if we look at a very small area that's protected inside a reef, and we have the flats, and we have the slopes, and we have the notches, and then we have the fills in between, then in that particular zone, we do see something that kind of resembles discretization, but it's not to the level where you can take a ruler and draw a line. It's unless you're looking at this from a very high perspective, and then your width, the width of your line is thick enough, then you kind of can draw those as discrete zones. But when you're looking at them very closely, they kind of bleed into each other. So I think the living assemblage is still a bit fuzzy, and that's something we see in ecological data. But adding to that the hydrodynamics, the redistribution of the grains, yeah, that adds another level of fuzziness. And then down the line, where we actually get to, we put our hand in the sediment and we pick up a pile of grains, then this differentiation is already kind of dissolved. The one exception for this, and this is kind of important, is when we look at the transition from a framework to a grain-dominated environment, because those are the only clear, distinct lines we can find. But in the one case where I actually looked at the example, the grains next to that were made from the material that we're building the framework.
Okay. Okay, so there's one more question before we can wind up for the day. So Vincent asks if what controls facies variability? Is it environment or tectonics? Are facies changes driven by sea level, subsidence, or faulting? And can you make a GIS connection too?
It's a little bit of everything. We've looked at we have this database on GIS, and you can, by the way, you can download the earlier iteration of the database and look at this. We've looked at buffers from distance from shore, and I looked at the integrate and some of the satellite data into that. The tectonic domains, I would not say are that significant in controlling facies, except for the fact that the tectonic domains give you wider shelves or narrower shelves, which affect what you would find, which not necessarily affect what you're finding them, but as much as how much you would find them and how steep is the gradient. So if you go to the Australian shelf, the gradient is, or the Persian Gulf, then the gradients are not as steep and you have a lot more smearing. And if you go to the Red Sea, then the shelves are very steep and the gradients are a lot sharper because of that. Temperature factors in, nutrients factor in, upwelling factors in. And this is just looking at it superficially, because once we start going into deeper waters, it gets a lot more interesting, because certainly internal waves domain begin to factor in, nutrient and light availability becomes a lot more imperative, because if you're in an area that has slightly more nutrients, then the light transmission through the water column becomes a lot reduced. So the zonation can become a lot more compressed, because you're going into the euphotic zone faster. Yeah, and again, distance for breathers, we spent a lot of time looking at this, and I think we're far from getting through all the options for the various control parameters.
Sure. I do not see any questions, and so I think we'll slowly wind up. Thank you everyone for joining this talk and for the great questions. And before we wind up, I would like to remind you all to join us next month for the SETS Online Student webinar, which is on AI, machine learning, and big data and sedimentology. See you then.